Hey Beaklings, here we go. We're going to dive a little deeper into photosynthesis and actually see how these processes work. Okay, here we go. So this presentation is a little different from our normal approach. Okay, you've already explored the various reactions of cellular respiration, so you should have a formidable background for learning the reactions of photosynthesis. Follow along with me or individual, jot down any notes to aid your understanding in the many graphics that follow. So this is what I present to my students when we're in class, and if you're watching this at home, all I'm asking you to do is try to follow the video and make sure that the words that I'm saying make sense to you. Okay, and if you need to jot down any notes, please do so. So normally I have a whole bunch of words for you to copy down and think about, but today we're just going to talk about it, and that's because we spent a whole chapter on things like electron transport, oxidation reduction. Okay, so hopefully this works out. Here we go. All right, like I said, it's always important to know where you're at when you're talking about these reactions. So where we're at is obviously inside of a plastid known as a chloroplast. Okay, and we're talking about being inside of the cells of the mesophyll, inside of the leaf of a plant. And remember, chloroplasts have three membranes actually. They have this outer membrane and inner membrane, and then they have the thylakoid membranes. Okay, and these membranes are like folded sacs. Okay, they're flattened sacs. And a stack of these things is a granum. Okay, and if you remember, we had one of my favorite scientists of all time, Lynn Margulis, talking about the endosymbiont hypothesis. Well, don't forget, these things, just like mitochondria, have their own DNA. They reproduce independently and things. Their DNA kind of looks prokaryotic, right? So this thing probably used to be a bacterium that millions and millions of years ago got taken in by another cell, right? So pretty neat. And what else should you know here? Well, basically, these are your solar collectors, okay? So the pigment, chlorophyll, is located inside of here, okay? So the light reactions are going to happen along these thylakoids. And what else should you know is that outside of the thylakoid, so inside of both of the outer and inner membrane in this area, but not inside the green, that's where the stroma is, okay? And that's where you're going to actually make sugars in the Calvin cycle. All right, next up, we should review what a photosystem is. So within the thylakoid membrane, instead of just having chlorophyll molecules, we have these protein complexes with the pigments mixed in called photosystems, right? And last time we talked about how photons, or light energy, hits the chlorophyll molecules, okay, and it gets transferred around through pigment to pigment, but eventually you're going to reach a special set of chlorophyll A molecules. And what happens here is these the chlorophyll molecules give up an electron, right? The energy is used to excite this electron, and now instead of being at the ground state, it's at an excited state. And the beauty of a photosystem is that the electron is not allowed now to fall back down and release heat and light, because you have this primary electron acceptor up there. Okay, so a photosystem is a way to harness the sun's energy. Rather than just jumping an electron and having it fall back down, we jump an electron and we grab it so that we can pass it on to an electron transport chain. Okay, so this next picture, I love this picture. I think whoever designed this is awesome. And sometimes I debate this. So sometimes I show you this picture as I'm doing right now, and sometimes I'll show you the Z-scheme of the actual photosystems inside of the thylakoid first, and then come back to this picture to make sure it made sense to you. Okay, so in other words, after we get done our next picture, you might want to rewind the video and look at this one again. Okay, and as you're looking at the next picture, scroll back and look at this one. Okay, and make sure that they go together very well. But pretty neat stuff here, because as you can see, we start with photosystem 2. Okay, and I told you last time, that's just, that's how biologists are. They discovered this photosystem first. So that's called photosystem 1. And then, oh, we discovered this one. But actually, in the Z scheme of photosynthesis, Z meaning up, down, up, okay, this photosystem always comes first. So rather than rename this to photosystem 1, they just said, the heck with it. Okay, so what happens here is sunlight is going to energize an electron from photosystem 2, okay, and remember this is actually at P680, or those special chlorophyll molecules you'll see. So you jump an electron, water is going to replace it, that's why they're blue, okay, and as the electron gets jumped, there's a primary electron acceptor up there. What was the whole point of jumping the electron for the first time? As you'll see, we're going to pass it down an electron transport chain, and we're going to pump hydrogen, really cool, into the thylakoid space inside of that thylakoid membrane. And by pumping the hydrogen in, it's going to be allowed to diffuse back out through what enzyme do you think? Just like last time, ATP synthase, right? The hydrogen or the protons are going to flow back out right? Facilitated diffusion through an ATP synthase, and that's going to generate ATP. We just got one of our high energy products of the light reactions. We took in sunlight and we made ATP. Now, where is our other high energy product going to come from? Well, as the electrons fall down the chain, they give up the energy to pump the hydrogen, right? Now, there's more sunlight coming in at photosystem 1, and you're going to nail it again. You're going to excite an electron. And this time, your primary electron acceptor sends it to a mini electron transport chain. But notice he doesn't allow it to fall back down. It's still a high energy chain. And here comes this chapter's little thief, NADP+. And NADP+ comes in there, okay, and it oxidizes the proteins of the chain, okay, and then it gets reduced to form NADPH. We now have our second high energy compound of the light reaction. So we got our ATP, thank you photosystem 2, which comes first, and then we got our NADPH, thank you photosystem 1, which comes second, and now we can power the Calvin cycle. All right, but before we do that, let's take a look at how this actually happens. Oh baby. So as you can see, this is a pretty cool and complex process. So here is the green disc, okay? This is the thylakoid membrane here. And as you can see, just like last chapter, you're actually pumping the hydrogen into a tighter space, right? This would be the stroma out here. The stroma is where it's a little lighter, okay? And the thylakoid space in here, that's actually right inside of here, okay? So what's going to happen? Let's go through the whole process again. Here comes light energy. All right, so photon energy hits the pigment molecules, and it passes the energy to a special pair of chlorophyll A molecules, and these guys are named P680. And like I said, P680 is amazing because it's maybe the strongest oxidizer on planet Earth. Because what's going to happen is, yes, you jump an electron up, okay, at photosystem 2, so the energy gets transferred, all right, and of course this whole thing increases entropy. Okay, but the electron or the energy gets transferred, the electron jumps up, and the primary electron acceptor will grab it. But here's the problem. You can't just take an electron from P680. You can't just take chlorophyll A's electrons and think that that's okay. Water now comes in, and water is going to get oxidized, thus reducing the chlorophyll A, and you're back to normal again. Aha. But water's electrons were low energy, right? This was formed at the end of an electron transport chain, perhaps. So that's low energy. That's why you need the sunlight to energize these low energy electrons from water, which become part of chlorophyll A, the P680, and shoot it up thanks to sunlight. Now what happens when you break apart water here, when you oxidize water? Well, you give off O2 gas, right? And you give up protons. How cool is that? Because you want protons down here. Now, why in the world did we jump this electron up? Well, we're going to pass it to an electron transport chain. And just like last chapter, these all have names, these different proteins and non-protein helpers here. And as you're passing the electrons along, they're going to give up energy. And what is that energy used for? Haha, you're going to pump hydrogen down. Okay, and the hydrogen, and I say down here in this picture, it could be up, it could be sideways, but you're going from an area of low concentration. This is out here in the stroma, right? This is like the big liquidy part of a chloroplast. And you're pumping it from an area of low concentration to an area of high concentration. That's hard to do. That's active transport. How did you go from an area of low to high? Well, you used the energy of the sun. Pretty neat. And as you pumped so many protons in here, so many hydrogen ions, guess what? That high concentration is a form of potential energy. And how are we going to tap into that energy? Well, we have this beauty again this chapter. ATP synthase, you're going to allow the hydrogen ions to diffuse through, and when that happens, this spins like a turbine around and around and around, and that's going to open up the catalytic sites or the active sites for adenosine diphosphate and inorganic phosphate to come together to form a bunch of ATP. Pretty cool. Now, okay, so there's our ATP. Why do we jump the electron again? As the electron came down the chain, remember, it's low energy, but then it gets high energy. And as you go down the chain, it's back to being low energy. So why do we jump it again? Why do we have photosystem 1 here, which comes second, and we, transfer the energy, and we come over here. This is called P700 now, these chlorophyll A molecules. And we jump it again? Well, because now, instead of allowing it to fall back down, we put it on kind of like a mini chain here. Okay, there's a little enzyme here, NADP plus reductase. And what's going to happen is NADP plus is going to get reduced by the high energy electrons, right? And notice that they're blue because they came originally from water. And if you're really good, you remember last chapter, as we were moving the electrons around, they were blue. That's because they're about to end up in sugar. And we started with glucose last chapter. Mind-blowing, right? So now, off of this mini electron transport chain, boom, we reduced NADP plus to get NADPH. And now we have our two high energy products, right? ATP and NADPH. And the beauty of it is, look where we created these products, not inside the thylakoid space, right? We don't want to be in here. We want to be out here. We want to be out in the stroma. Because what happens in the stroma is the Calvin cycle. And what powers the Calvin cycle? Well, not sunlight, indirectly, right? It's sunlight, but directly it's powered by NADPH and ATP. And it's ready to roll. So here we go. Okay, so the Calvin cycle, this reduces CO2 to sugar, which is powered by ATP and NADPH. That's all it is, right? Let's get some CO2 in here and turn it into a sugar using the high energy from the sun, basically. Okay, so we just covered this part. We just covered the light reactions, and we just got our high energy products, and now we're going to use those high energy products to power this Calvin cycle to turn CO2 into a sugar. All right, well, how does this happen? First of all, the first step is carbon fixation. So CO2 comes in. Let's say 3 CO2 come in, okay? And again, all of these intermediates have names and things, so you end up with something called like phosphoglyceric acid up here. Actually, there's a short-lived intermediate. It gets transformed, and not important whatsoever. What's important is to understand, oh, okay, let's take a look here. RUBP, or ribulose biphosphate, is kind of the beginning and the end of the chain, just like oxaloacetate was last chapter. So RUBP is sitting here, and if you count 1, 2, 3, 4, 5 carbons, but there's 3 of them. All right, so get out your calculators, use your cell phone if you have to. Let's figure out how many carbons that is. 3 times 5 is 15. All right, good. And now here comes 3 more, right? So 15 plus 3 more, okay? And what do I mean plus 3 more? We're going to incorporate this carbon dioxide in these molecules, and now we're going to have 6 PGA, or this molecule right here. Okay, and that's going to be 6 times 3 is 18. 15 plus 3, 18. Okay, so we're still good with our carbon, right? And now this molecule right here, all right, we're going to energize it. We're going to say, well, CO2's now been fixed in there. All right, let's go ahead and blast this thing with energy. Let's try and make a carbohydrate. So basically what's going to happen is ATP, thank you very much. Okay, thank you, light reactions. We made ATP, and we're going to actually invest 6 of them right here. Not only that, but step 2 is reduction. We're going to go ahead and take the high energy electrons that we jumped at photosystem number 1, right, the second photosystem. We stole these electrons, right, and we're going to go ahead and reduce the molecule, okay, or NADPH gets oxidized to form NADP+, and now we have G3P, or glyceraldehyde 3-phosphate. This is a sugar. It's basically half of glucose. If you go back and look at our drawings for glycolysis last chapter, when you broke it in half, you'd see that they're called G3P, right? Pretty neat how this is all connected. Okay, so now here's the thing. You still have 18 carbons, and now you have 6 of these beautiful sugars, right? So can we just use all 6 sugars? Well, theoretically, sure, go right ahead. But the problem is you have to reconvert some of them or regenerate your RUBP. So if you took out all 6, you wouldn't be able to keep the cycle going. So what happens is, right, 1 sugar can be your product of the Calvin cycle. So we're going to be a net loss of 3 carbons here from the cycle. And what does that leave as? G3P. And that G3P, of course, can become glucose or any other compounds that you need it to be. All right, and there you can see it coming out. So 3 CO2 come in, so 3 carbons coming in, 3 carbons coming out in the form of G3P. Now what do we do with the remaining 5 sugars? Okay, the 5 glyceraldehyde 3-phosphate. Well, you go ahead and regenerate RUBP. Now how do you get from this? Now 5 times 3 is 15, 15, and then 3 times 5 is 15. So how do we regenerate? Well, some more ATP. All right, so thank you, sunlight, for allowing us to generate that ATP inside of a chloroplast. Okay, And now that you have your RUBP regenerated, hey, as long as that plant has more CO2 pouring in, we're ready to go another round, and we're just going to keep stealing sugars. Pretty amazing. So a plant actually used 3 simple things, right? Sunlight, water, and CO2, and you were able to make sugar out of it. That's pretty awesome, right? And that feeds the world. So we depend on the sun for basically all of our energy, and we depend on these photoautotrophs for all of our food. Right? Pretty cool. Now, one guy that I didn't mention too much during that cycle was right here. He's an ancient enzyme named ribisco. And he might have been on planet Earth since before oxygen even showed up, which is crazy. So ribisco is my favorite enzyme, actually. And he's a big part of this Calvin cycle because he's going to help the CO2 get fixated into the RUBP or ribulose biphosphate. But there's some trouble here. Other methods of carbon fixation have evolved in hot and dry climates. Okay, so we'll talk about what plants have done to kind of protect themselves. But one problem that happens because of usually hot climates is photorespiration. Okay, this occurs in the light and consumes oxygen and releases CO2. Wait a minute, consuming oxygen and releasing CO2, that's like respiration, right? That's not this chapter, that's last chapter. But unlike cellular respiration, it uses ATP instead of producing it. Oh no, right? So we're not even getting the ATP yet, we're using ATP, and unlike photosynthesis, it actually yields no sugar. So photorespiration can in fact drain away as much as 50% of the carbon fixed by the Calvin cycle. Okay, and a lot of this is because ribisco, that enzyme I was just telling you about, has an affinity for oxygen as well, right? Ribisco's sitting there and he's saying, hey, I would love some CO2 right now, but if he's overloaded with oxygen, and we'll see why that could happen, okay, but if he's overloaded with oxygen, he'll take in oxygen, right? And then there's no carbon fixation, there's no sugar being produced, right? No good. It's kind of like an evolutionary boo-boo or an evolutionary relic that's left over. All right, remember, we're not perfect, nor are plants. Well, I'm perfect, but not exactly. So you can see here, these are the stomates, right? Here's stomata of a plant. And I told you there's these beautiful guard cells on the leaf that can open and close. Okay, the openings are the pores. And basically, if it's open, that means that gas exchange is happening. That means that, you know, what's going into the plant, a whole bunch of carbon dioxide, and what's coming out is a whole bunch of oxygen. Now, a lot of kids will ask, wait, don't plants have mitochondria too, and don't they need oxygen? They sure do, and that's a very good point. The thing is, we're huge, and we have big muscles and big brains, right? We need a ton of ATP. So cellular respiration for us is a big deal. Do plants eat sugar via cellular respiration? Sure they do. They just don't need near as much as we do. Okay, so a lot of that oxygen that they produce, there's so much of it, they just release it. And if you remember last chapter, oxygen being released into the atmosphere was catastrophic when it first started happening. Oxygen is a deadly, poisonous, caustic gas. It's so electronegative, it probably makes us age. It might cause disease inside of us, right? So that's why antioxidants are very important. You don't want a whole lot of oxygen around. But here's a process where, thank you, plants, because we need oxygen to live. Without oxygen, we can't make ATP. We're in big trouble. Okay, so it's such a weird thing how this evolution has happened with these processes. Anyway, this stomata right here you can see is closed now. Okay, now think about what that means. If you have a closed stomata, that means that no CO2 is entering and no oxygen can escape. Oh, and that brings us back to this photorespiration business, right? So if you're closed, let's pretend both of these are closed and the entire plant is currently closed for business, you have some CO2 in there, I'm sure. You have some carbon in there, but once that runs out and you produce all that oxygen, but now the oxygen doesn't have a place to go, right? You split a lot of water still, so now the oxygen might be sitting inside of the plant and ribisco is going to start grabbing that. Okay, so photorespiration is basically a waste. Okay. So how did plants evolve to avoid photorespiration? Well, we have 2 types of plants here, and they're really, really cool. We have C4 plants and CAM plants. Okay. Now, we just went through the Calvin cycle, and if you remember, there were 3 carbons coming in and 3 carbons going out. Okay, that's a C3 plant. Most of the plants around are C3 plants. But when you get into these specialized plants that live in, you know, hot or arid climates, they had to have a way to say, hold on, if I keep my stomata open all day, I'm just going to have transpiration happen, which is the evaporation of water from their leaves. They're going to lose all their water and they're going to die. right? Because photosynthesis certainly needs water. So what certain plants do is they say, all right, I'm going to go ahead and shut my stomata, okay, when it gets too hot so I don't die from losing water. But the problem with that then becomes photorespiration, and oh no, what does a plant do? Right? Well, certain populations of plants, certain species have evolved these really cool backup systems here to protect themselves. For example, a C4 plant, what happens is CO2 comes in, and it's in this mesophyll cell, okay, and you actually store it as a 4-carbon compound, okay, and then when you need the carbon dioxide, okay, you have it stockpiled. For example, if it's a hot day and this plant decided to close its stomata down, and you're like, oh no, I'm out of CO2, well no you're not, because in your mesophyll cell, you have a whole bunch of 4-carbon compounds formed, which you can certainly, okay, hydrolyze and get CO2 out of them. And then the CO2 comes into what we call the bundle sheath cell, and you can keep photosynthesis going. So it's a really, really cool way for plants to keep going, okay? A good example of a C4 plant would be sugarcane, right? So they're smart enough to close their stomata, but they hopefully have some carbon stored up so that our Cheerios don't taste disgusting. Okay, and over here you can see this is a different type of system here, because in this one, in a C4 plant, you use different cell types, okay? Now over here what you're doing is different parts of the day, okay? So this plant's so smart, all right? Must have a great big brain, because what happens is the CO2 pours in at night when it's cooler, okay? You don't have that hot sun beating down at you at night. So this plant is going to, and CAM, all right, cressolation acid metabolism, these plants open up their stomata, okay? The CO2 pours in at night, and then during the day, sure, you're going to close that up, but that's okay because you stockpiled so much CO2 in here again as 4-carbon compounds and CO2 that when the sun comes up and it gets very hot, okay, the stomata are going to close, but that's okay because here's all the CO2, and now the light reactions kick into high gear. They're producing your ATP and NADPH, and that's okay because here comes CO2, and we're ready to keep going through the Calvin cycle. So a very cool evolutionary adaptation. Over here, we have different cell types that help the plant if it has to close its stomata. And over here, we have same cell, but different times of the day, it opens and closes its stomata. Pretty cool stuff. Okay, and one last thing today, something you've been learning about since you were very young, hopefully, is climate change. All right, is the direst issue facing our planet. So we are in big, big trouble on our planet because we have a whole lot of greenhouse gases. We're not taking care of our planet. We're making decisions, okay, whether it's nationally or globally. We're doing things that are actually making this problem worse instead of better. And there have been some administrations in our country and others that have done a lot of really good things when it comes to climate change, and there's been other administrations that are really setting us backwards. But the thing is, scientists all agree this is a huge problem, okay? And a lot of it is man-made. Now, climate change is not, okay, it's not only like, oh, this is happening now and that's it. No, climate change is a natural process. That's happened many times. Like, we've had ice ages before, okay, and we've had periods where the Earth is extremely hot before, okay, but the thing is, those are natural progressions and they take a very long time to change. What's happening here is, well, we're rapidly heading into a very hot period of our Earth, and actually this is probably going to cause a mass extinction event on our planet. Right? Life as we know it won't be the same. And you might be thinking, well, it's not going to affect you. No, depending on your age, it probably will affect you. And it's already affecting you. It's already affecting all of us. Okay? But not only that, think about your kids, their kids. Right? Are we still going to have a planet that's habitable? Okay? Are you still going to have all of these places to live that we currently do? Are these new weather patterns and things like that going to destroy what we used to know and love? Are the sea levels actually rising? Will there be glaciers left? Right? How many species will be gone forever? All because of things like pollution, okay, and us not maybe like eating enough plants instead of meat, right? Agriculture, and there's ways to do things smartly, right? Look at energy, for example. We have renewable resources out there. Let's use solar, let's use wind, right? Hydroelectric power, right? Not burning coal, for example, right? Those days should be behind us, and we're getting there. Right? And most major developed nations understand that this is a huge problem and we're all working on it. So it's scary stuff. And as you can see here, basically what happens is sunlight comes down and hits the Earth, and it's always going to bounce off, okay? And some of it escapes in the space, okay? And some of it gets redirected back to Earth. This is common, and this has always happened. The problem is, as we have more and more and more of these greenhouse gases in our atmosphere, the sunlight comes down, but instead of being able to jump back up, okay, and reflect back out into space, some of it escapes, but a lot of these greenhouse gases are trapping that hot energy, that solar radiation, and it's trapping it within our atmosphere, okay? And because of that, well, sure, the Earth is getting warmer. There's no doubt about it. So what can you do personally to stop this? Well, you can educate people. You can educate yourself, right? You can listen to scientists, right, and not sensationalists. And you can also do things like, sure, carpool. Let's get efficient cars out there, right? I currently drive a great big pickup truck. There's no reason to do that. What, to go to the mall, right? I don't need a big pickup truck most of the time. Plant trees, right? That's one thing my family does. Since we moved in here, we have like 8 or 9 new trees in our backyard that we didn't have when we moved in. So we keep, our population of humans is exponentially growing, and we keep deforestation going on, right? We're losing a lot of what helps resist climate change. So you have to think really hard about this stuff, because we only got one chance at keeping this planet in order. Pretty scary stuff. But anyway, so that's a little bit about photosynthesis. We dove in a little deeper today, and I talked about how the processes actually work, right? And hopefully this all makes sense. Now, if you're watching this video thinking like, wait, I don't really understand exactly how this is happening and what redox is and oxidation. Well, yeah, we already spent chapter 6, right, or cellular respiration, talking about all that stuff. So really all this is the reverse process of cellular respiration. Anyway, I hope that video is helpful, and take care.